So welcome all the participants who register and join us for the webinar of the eLife. We are focused on the latest research in biochemistry, particularly focusing on proteins. Today we'll try to make an exciting presentation about unpuzzling secrets of proteins. And I'm Ivan Dikic, eLife Senior Editor, and would like to really welcome our guests as well. And we have 4 panelists today, and among them Manu Hedge from LMB Cambridge. Then we have Anne-Claude Chengras from Lunenfeld Research Institute in Toronto, Wade Harper from Harvard Medical School in Boston, and myself from Goethe University in Frankfurt. 4 of us will try to present in 10 minutes different topics, and the topics will range from making proteins, how translation and folding works. That will be Manu's presentation. Then we're going to have interacting, connecting proteins, and this is Anne-Claude topic, and then monitoring proteins, particularly on a quantitative side, utilizing proteomics. Wade Harper is gonna give us his view on this area, and finally I'll finish by degrading or removing proteins. And each of us will have around 10 minutes. Will be around 10 minutes presentation, and there will be at the end 20 minutes of a discussion, and we would like very much that discussion is active. There is a pool where you can leave the questions. Questions will be pulled together by the staff of the eLife, and they will be then presented to everyone who received the question. And we really hope that this last 20 minutes will be quite an active area as well. So thank you all for participating. Thanks panelists for joining. And now I think Manu will kick off the webinar.
All right, well, yeah, it's a pleasure to tell you a little bit about making proteins. If I could share my screen. So I'm going to tell you a little bit about the most active areas, at least some of the most active areas in the area of translation and protein maturation. Now, translation has been a major area of interest for a number of reasons. The main one is that this is the last step in gene expression, and proteins carry out the majority of biochemistry inside cells. And so one really wants to understand at any one given moment what all of the proteins that a cell is making at any given time and what all the proteins are that are present inside of a cell, because that essentially determines the identity of that cell and what it's doing. Now, cells have many billions of proteins, and actively dividing and actively metabolizing cells are typically making several millions of proteins at any given minute. And so what we really want to understand then is how this process works and what cells are, what protein cells are making. So the process of making proteins, translation, is really, I think the chemistry of this is understood through biochemical and structural work over the past many years. And because the process is very well conserved, work that's been done primarily in bacterial systems has really led to a great understanding of the chemistry of translation. But translation continues to be a very active field for a number of reasons. First of all, it turns out that translation is highly regulated, and particularly in metazoans, translation is regulated during development, during changes in environment, and is misregulated in disease. The second is that there's an increasing appreciation that translation is very tightly coupled to the maturation of a protein. So the processes of folding or modification, localizing the protein to the right part of the cell, and assembling it with other polypeptides in order to make a functional product. Now, these processes are also very tightly controlled in terms of making sure that the proteins that are made correctly, and that's a process called quality control. And both the regulation of translation on the one hand and the maturation and quality control on the other are increasingly appreciated to play roles in disease, and I'll get to that in just a moment. So for these reasons, translation continues to be an extremely active area of research. Now, what one wants to know are a couple of things. One is we want to understand at a global level what the proteins are that are being made in a cell at any given time. And at a mechanistic level, we want to understand how these processes are regulated and how they actually work in more atomic and molecular terms. And these are really the two broad areas. And both areas, I would say, are being driven by advances in technology. So if we want to quantify all of the things that are being translated, classically, the best way to do this, if you look at this hypothetical cell, is to isolate the ribosomes or the polysomes from these cells, and then sequence all the mRNAs that are associated with them. So this originally was done using microarrays, for example, but more recently using RNA-seq. And really in a very nice advance by Jonathan Weissman and Nick Ngolia, they realized that if you digested such a sample with nuclease, you could then extract the small pieces of RNA that are protected by the ribosome. And when you sequence these, really get a snapshot of where all the ribosomes inside of a cell are sitting on the messages that they're translating. And so this process, which is called ribosome profiling, has really started to allow the quantitative analysis of translation inside cells. And it's quantitative because the number of reads that you get from particular footprints is a measure of how many ribosomes are engaged in translating that particular sequence. Now, this provides, of course, a very nice snapshot of cells in culture or yeast, but there are more sophisticated questions that I think this is starting to be applied to. For example, suppose you want to know during development all the things that are translated in a particular tissue. So this is a picture of a developing Drosophila embryo, and the green cells are developing salivary glands. And so one strategy that people are starting to use is you can, for example, label or tag the ribosomes that are expressed just in these cells. And then if you use this for isolation, you can then use RNA-seq or ribosome profiling strategies to try to get an understanding of the kind of translational regulation that occurs at different steps during development. Of course, there are issues with respect to how strong the signal to noise is, especially if you're dealing with a very small number of cells, but I think this is one of the challenges moving forward. Because ideally, you want to really understand not just translation in unicellular organisms such as yeast, but really in complex metazoans. And in that case, you need this spatial information. The second aspect of spatial information that I think one would want is within a cell, you might want to know where translation is occurring. So one example of this is translation that's occurring at a particular organelle. For example, in this example, the endoplasmic reticulum. And so one strategy that's been developed to do this is if you, for example, express a biotin ligase at the location that you're interested in, you can then tag all of the ribosomes that happen to be at that location. And that provides a molecular handle to isolate those ribosomes, sequence those messages, and see what's being translated near the endoplasmic reticulum. A variant of this, for example, is if you specifically isolate ribosomes that have particular factors bound to them, you can then ask what are the mRNAs that are engaged with that factor or proteins that are engaged with that factor during their translation. So these kind of selective methods provide a different level of spatial information. And ultimately, I think the kind of questions one wants to ask is very sophisticated ones. If you take, for example, this neuron, there's translation occurring in different parts of this highly polarized cell type. So for example, if you look at the dendrites, which is circled here, you might want to know what's being translated specifically in those dendrites, and how does that translation profile change when, for example, the synapse that's at that dendrite is stimulated, and how does this change over time? And so, as I said, I think that issues of specificity and signal to noise really have to be solved before one can address these questions in great depth. But these are the kinds of questions that I think one wants to address. So if we step back, what you really want to know is at single cell sensitivity, ultimately, what is the profile of all the proteins that are being translated? And ideally, you'd like very high spatial as well as temporal specificity. And if you had these kind of tools, which are actively being developed, you can then apply it to physiology, development, and various disease processes. Now, once you have a kind of description of all the messages that are being changed during development or some process of interest, parallel methods are being developed for being able to watch translation in vivo. Now, currently, these methods involve really rather large tags, but again, improvements in being able to watch translation in real time and watch how it changes, I think, is one of the real challenges moving forward. Now, all of this would provide, I think, a description of what happens, but ultimately, I think one wants to link that to specific mechanistic events. And that's really, I think, the second broad area. Translation, in particular the ribosome, receives many inputs. So there are lots of factors, dozens to perhaps hundreds of factors that bind the ribosome, many modifications such as phosphorylation, changes in tRNA levels, and so on. And all of these things impinge on translation in order to control various steps in the output. So of course, we know that initiation, elongation, and termination are controlled, but other processes such as targeting of the ribosome to particular parts of the cell, decay of the mRNA, protein modification, and so on, are all controlled by ribosome binding factors. So these questions, of course, are addressed classically using biochemistry, genetics, and so on. But one method that I think is really having a major impact here is cryo-EM. And this is in part because this method allows one to look at very large complexes, and the ribosome in particular is a great target for this because of its very high contrast in EM. And you can look at things that are bound to the ribosome and in fact, get sufficient resolution to build atomic models. So in this particular example, you're looking at a ribosome bound to one of the factors it associates with, a protein translocation channel. And this allows one to then understand how these partners that bind to the ribosome actually function in molecular terms. One can even do this in more native samples using a process called cryotomography. So in this example from Friedrich Forster's group, ER, just whole ER, can be analyzed by cryotomography to see what the components that are associated with membrane-bound ribosomes look like. And this is more moderate resolution compared to the single particle EM, but again, enough to start giving you a flavor of the molecular functions of these factors that bind. So in this area, really, I think what we want are all of the factors that directly impinge on translation. And we want to understand the mechanistic basis by which selectivity is determined, because of course, much of translational regulation is selective. And we ultimately want to coordinate that with cellular physiology and see how this changes during disease. And again, very subtle differences in translation can have significant impact during processes such as neurodegeneration or neurodevelopmental disorders. And so I think understanding translation both in global terms, as I mentioned at the beginning, and mechanistic terms, I think is where some of the really important questions lie. So with that, I'll stop and hand over the baton to Anne-Claude.
All right. Start the presentation. So thank you very much, Manu. So once you make those proteins, you have to get them to perform their activities, and that, as everybody knows, involves these proteins interacting with one another to perform their function. And so my topic is about interaction network. But before we go into networks, I just want to remind you that there are different types of protein interaction in the cells. So for example, I show you the ribosome that Manu was talking about, where we have a really stable, well, not easy, but biochemically purifiable complex, really a molecular machine where all of the subunits are held together with fairly strong interaction that involves a lot of buried surfaces. In contrast to that, we have things like signaling pathway. I'm showing you an example from some of our recent work where we have multi-step activation of phosphorylation-mediated low affinity binding events that ultimately will result in this case, the activation of a specific kinase that's going to be then going on and modifying its substrate. So you can imagine that if you have such different type of interaction, and we can generalize that to the rest of the cells, so what you're going to be dealing with is a mixture of transient interaction that are in general regulated, for example, by post-translational modification, and permanent or more molecular machine type of interaction. So this spans the wide range of affinities and binding properties, with on the one side interaction often mediated by short linear interacting motifs such in signaling pathway, and those will create interaction that are easy to remodel to affect an activity of change in binding. At the other hand, we're going to have our molecular machine. So all this to say that really what we're dealing with is a number of different, completely different interaction, and one technique to study those interactions does not fit all, right? So so just before I get into any of the technique, I just want to remind you what we're trying to do here. We're trying to detect and report interaction between a protein A and a protein B, which normally we would call bait and prey, solely based on which one we capture and which one we detect. And those interaction methods, in general, we split them into two different classes. The interaction monitoring approach or binary approach require you to have both the knowledge of the bait and the prey, so that what you're going to be trying to see is capturing the bait and seeing if the prey came together. And fundamentally different to that are those forward screening approach or NRE approach, where only usually the bait is known and the prey is to be identified usually by mass spectrometry. This can also be used to identify post-analysis both of the bait and prey. So you may not need to have any knowledge, certain knowledge of the bait and the prey. And what I mean by that is that if you have a protein, this bait protein A, what you're going to be able to capture is a number of different interactions. Some of them are going to be direct and some of them indirect. Just to come back to the interaction monitoring approach, those have started really with the yeast to hybrid system that's in field developed now many years ago, but there's a number of different approaches that vary in what they're monitoring. So again, like some of them, like the yeast to hybrid, will be based on reporter gene activation in living cells, so they do not require cell lysis. They can essentially monitor and give you a readout in live cells. But there are some other variations of the approach that are quite interesting. For example, you have localization-based approach that can inform not only on the fact that you have an interaction, but also where this interaction occurs inside the cells. And you have a number of different biochemical approaches. And again, this is a really growing list. Almost every week, I'm an editor at Molecular and Cellular Proteomics, and we get a paper that develops or improves one of those techniques. I'll just comment briefly on some of the advantages and drawbacks of some of those approaches, using as an example the yeast to hybrid that's by far the most widely used. So the big advantage of the yeast to hybrid for network biology is low cost and ability to make it high throughput, especially now as barcoding is possible. Another advantage, as I said, is you don't need cell lysis. So you can capture interactions that are intrinsically fairly weak, right? Because you don't need to maintain those interactions. On the drawback, of course, this is a fairly artificial system, and it's kind of hard to detect interactions that are mediated by post-translational modification. It's also hard to detect protein complexes, and you need also, you lose also your spatial constraint. Despite this, and even though for many years, it was thought that the yeast hybrid yielded a lot of false positive, there's been a huge effort, especially by the Vidal lab, to essentially try to calibrate the assays by running both positive and negative reference set on each of their experiments, so that they can have an idea of which strength of signal actually correspond to an overall good positive interaction. So this is a system they've used intensively. So the Vidal lab has built a collection of so far 18,000 different yeast to hybrid open reading frames. So they're testing them for binary interaction in a matrix format. And so far they've reported over 80,000 interactions. So this is really, really powerful. This really is a very important contribution to network biology, and also a lot of the tools that have been developed to analyze networks have been coming from this yeast to hybrid. Now I want to spend the rest of the time talking more about the forward screening approaches. This is of course what I do, but I think it's also where there's been a lot more advances recently. So those can be split, at least for this presentation, in four main categories: the affinity purification coupled to mass spectrometry, which is a more classical, and three emerging techniques: protein correlation, profiling, proximity labeling, and chemical crosslinking. And what they have in common is the identification will be by quantitative mass spectrometry, which Wade will discuss in a minute. So just to come back a little bit to the most classical approach, which is this affinity purification coupled to mass spectrometry. In this experiment, all you're trying to do is to capture from a cell lysate a bait protein alongside its interactor. So you're trying to wash away the rest of the cell, the contaminants, and you're coupling that to mass spectrometry. So the big advantages of that is this gives you almost near endogenous conditions, especially if your bait is expressed at near endogenous level. It's compatible with many systems and many species. You can also combine that with any kind of treatment to look at condition-specific interaction, and you can derive scores and some kind of stoichiometry. The main issues that we have here are the fact that this is actually requiring cell lysis, right? So you need those interactions to stay together during the lysis and the purification. So that introduces some bias in the cells. So weaker interactions tend to be lost, and interactions that occur, for example, in a membrane tend to also be disrupted in most generic protocols. This being said, this is extremely powerful. We use it, Wave uses it even more than us. And this requires you to develop tools to score your high confidence interaction, which different groups have done, including us. So we score, for example, the data, quantitative data from a bait against negative control using our tool Saint, and that really tells us which are the proteins that seem to be specific to our samples. Wade has developed back in 2010 now, or 2009, this compass method that enables him to look at something similar to what we do with Saint, but across an entire data set. And there are other type of tools that are complementary to that enable you to score in addition or instead based on the topology. So if you pick up reciprocal interaction or interaction amongst different members of a complex, you can either boost or penalize those interactions. So this together generates really high quality interactome, and again, like Wade as together with Steve Gigi led the field on that, using very much the same base that Mark Vidal has been using for yeast to hybrid. Is in the process of generating has generated the largest, and it continues to generate huge high quality interactome data from that, moving into different cells. So really in terms of large scale data set, this together with the Vidal yeast to hybrid data set really have opened a lot of research. So I'm going to talk just a little bit on the newer approaches. So the first one I'm going to cover is protein correlation profiling, which again starts from a lysate, but in this case, you're going to want to not tag anything, but get a global profile of everything that you're going to have in the cells in terms of complex. This is really trying to identify protein complex molecular machines. And the basic idea is just to use old classical chromatography, including size exclusion chromatography shown here, and essentially use a mass spectrometry as a detection to monitor every one of the fractions so that you can, based on the co-elution profile, reconstitute complexes. So this is really a global approach. This is not intended to be a target identification. And again, it has huge advantages. It enables you to rapidly look at big drastic changes in the interactome following a perturbation, for example, viral infection, with again, many of the same drawback as any biochemical purification, but also some major issue in dynamic range, which I think Wade is going to cover. This being said, it's been used by now several groups, but leading by Andrew Emili and Marcotte. This is an example of their paper where they use this approach to reconstitute protein complex across evolution. So really interesting technique. The next one is actually growing really quickly, and is globally called as proximity-dependent bioassimilation. The two most well-known approaches are BioID and Apex. They work essentially the same. You fuse to your bait of interest an enzyme that then mediates inside living cells pre-lysis the bioassimilation, the covalent bioassimilation of residues within this cloud of activated biotin. Again, like APMS, you can do that in near endogenous condition in many systems. You can use essentially the same score as you use for calling signal to noise over the background. It's useful for both direct and indirect interaction as well. But the big advantage here is you don't need to get cell lysis. So the interaction don't need to be maintained throughout purification. The drawbacks is that right now, this is fairly new. So right now it's still difficult to interpret the complex patterns of interactomes or proximity interactome that you're going to get. So you're going to get both direct and indirect interaction, but also localization information. And to entangle that is still fairly complicated. Still not clear what the dynamic range is, what the blind spots are, and how good these approaches are for dynamic interaction, even though there are newer enzymes that enable to do labeling in very short time frame. So I just need to mention that the size consideration is something that we need to look into more detail. So the size labeling radius is thought to be about 10 to 20 nanometer, although this is still debatable, which if you look at the ribosome, that's about 30 nanometer. That tells you like you may get essentially differential labeling of different subunit. But by and large, you should not use this as a molecular ruler. I won't go too much into that, but really there has to be more thought put into interpreting those data sets. And this is just an attempt that we've done in a recently published paper where we essentially try to infer a relationship between the on-tag endogenous spray using multiple bio-ID experiments, in this case 140 or so. So lastly, I'm just going to talk very, very briefly about the chemical cross-linking approach, because I think this is where a lot of work is being done right now. So by and large, this is something where you can treat cells or lysate or isolated complex with a chemical cross-linker that will fix a molecular ruler, for example, something that will be 10 nanometers. And then the idea is that at the end, you try to purify back those cross-linked peptide and identify to get the identity of both of the partners, which you can then map back onto the complexes and the interactors. So you don't need tagging. It's compatible with many systems. It's a global approach. You can do that pre-lysis if you do it in vivo. And importantly, that provides distance constraint to modeling. The big challenge there are really the depth of the sequencing and the issues of protein abundance and dynamic range. So as of today, in a good experiments, we get about 5,000 cross-links, which again are not independent evidence for 5,000 different interactions. But again, if you compare that with the current network biology data set, we're very far from being there. So right now, those approaches are better coupled with other approach, and we're doing the same kind of thing with BioID as a company. So to come back to the challenges, again, this is very similar to what Manu was talking about. We really need to get a better handle on how to annotate, discover, annotate, report, and visualize those contextual interaction, regulated interaction, and so on. We need a better handle on how to unify across method, because even though databases such as BioGrid or Intact record all of those interactions, it's very hard to analyze them globally, especially as much of the quantitative data is stripped from the database. And lastly, I think where this is going to be quite useful is we're going to need to move beyond those network view more toward mechanistic structure. And again, this will require the integration of all this information, contextual, method unification, to essentially join the data into better structural model and better mechanistic model. And this is just an example that was published using all kind of approaches, cryo-EM, mass spectrometry, histohybrid in a recent eLife paper. So thank you very much. And then I'll pass to Wade for the next talk.
Okay, so thanks very much, Anne-Claude and Manu, for fantastic introduction. So I'm going to follow on from, especially from what Anne-Claude was discussing, and focus to quite a large extent on this problem of dynamic range in understanding protein function. And so you've heard already about the ribosome, how proteins are made, and this generates a huge level of complexity. And in the next couple of slides, I'm going to talk a little bit about what the complexity means for the abundance of different proteins. And so we know that in cells, there is quite a large dynamic range of proteins. And on the right side of the slide, what this shows is basically the relative abundance of about 10,000 proteins present in a cell. And you can see that it ranges over about 7 orders of magnitude. So what this means is that for proteins that are highly abundant, those might be much easier to measure than proteins that are of very low abundance. So a few numbers are sort of interesting to think about. So how many protein molecules are there in a single cell? This is not exactly trivial to measure, and there are estimates that have been produced mostly on proteomic data, but the estimates range across maybe 2 orders of magnitude. And so this depends to a large degree on the cell types that are being examined, but also on the methodology. But the general estimates are that there's somewhere between about 10 to the 9 molecules per cell, up to perhaps as high as 10 to the 11 molecules per cell, or protein molecules per cell. So that's quite a large number, and it sort of boggles the mind to think about how all these molecules get made, how they get assembled. So that's to some degree what we'll be talking about. So if you look at just a standard HeLa cell, it might have 10 to the 9 proteins, but the top 40 proteins constitute about 25% of the total protein mass in the cell. So these are the very highly abundant components, but yet they are really sort of constituting the vast majority of protein abundance. Even the top 600 proteins is about 75% of the protein mass. So this gets you sort of thinking about the amount of protein that is in the sort of more lowly expressed proteins. So the least abundant proteins in the HeLa cell, the 50% of the least abundant proteins account for 2% of the protein mass or less. So it's a very small fraction. So many proteins are constituting a very small fraction of the total protein mass in a cell. This makes detection and quantification of particular events challenging in certain cases. Now, there's a different way to think about the number of molecules in the cell, and that's by looking at copy number per cell. And so in this slide, what you're seeing is that the most abundant proteins in the cell might have 10 to the 8th copies of themselves. Now, those won't all be identical. They'll be potentially modified in different forms and be part of different complexes. But if you're just looking at the individual protein, it could be in that order. So these are very abundant proteins, GAPDH, etc. On the range of 10 to the 7 molecules per cell, or copy number per cell, are things like the ribosome, which we've already heard a lot about. The nuclear pore is also quite abundant. So we're talking about 10 to the 6 copies of the components that make up a nuclear pore, and there's many different proteins that make that up. Then way down the scale, where you're talking about hundreds to maybe thousands of molecules, are more regulatory factors, kinases, transcription factors, and this protein SLX4, which is a component of a holiday junction resolvase, which is a very low abundance protein that nevertheless is playing key roles in controlling aspects of DNA repair. So if you think about copy number, what you begin to wonder about is, what does that actually look like in terms of protein concentrations in the cell? And so there's this very interesting analysis that I'm showing here in this little movie of what the protein cytoplasm might look like in a cell. And so you see many different protein assemblies in different colors, and those are having dynamic behavior in the cell, but they're very, very concentrated. And so this is going back to Manu's question of how you actually build proteins in this very complex environment, how they are properly put into the right location in the cell. So that raises the next question then. You have this wide array of protein abundances. How do you actually organize those into the different organelles? And so what you see here on this slide is an electron micrograph of just a standard cell, where you can see the plasma membrane, you can see the mitochondria, you can see the nucleus, the Golgi, endoplasmic reticulum. And what's pretty clear is that these different components, all these different molecules that you see, all the different protein molecules, are arranged in particular structures in the cell. These are often membrane bound, although they don't have to be. There's now a large amount of interest in understanding non-membrane bound compartments. There's also machines, and as Anne-Claude talked about, there's much more dynamic signaling complexes that are present. And so the question is, how can we analyze all of these different components in a quantitative manner? And so what has evolved over the last several years are a number of different strategies that let us examine multiple different aspects of proteins in cells. So at one level, you can use different kinds of enrichment strategies to look at organelles, protein interactions, as you've already heard about, and also protein post-translational modifications that include phosphorylation, ubiquitination, etc. And by combining those different enrichment strategies with various sort of labeling strategies, or in fact also label-free quantification strategies, one can use proteomics to basically disentangle this complex array of proteins that are present in the cell. I don't have time to go into all the different labeling strategies. There are different philosophies of how you go about doing things based on the experimental situation that you have, but the bottom line is that you're using various either pre or post isolation mechanisms and chemistry to quantify individual peptides, or in some cases protein amounts, using essentially the power of mass spectrometry. So I want to give you one example where we look at organelle enrichment, and this gets back to some degree to this sort of system that Anne-Claude mentioned, this sort of correlation profiling. But instead of looking at individual protein complexes, this is now looking at organelles. And so in a very nice paper from the Borner lab, they employed sort of simple fractionation protocols to separate organelles in a manner that allowed them to then do correlation analysis of when and where in these different fractions that they purified, different proteins appeared, and then quantify those by mass spectrometry. And the very, very interesting outcome of that is essentially a description of the relative abundance of proteins present in each different class of organelle. So you can see here that for example, the nucleus contained about 21% of all the proteins, whereas the lysosome contained only 0.2%. So you can already sort of tell that there's a huge distribution or difference in the distribution of proteins as you go across the cell and look at different organelles. So for example, roughly 6% of the proteins in the cell are present in the mitochondria. So this is actually sort of interesting to think about when you then layer on top of that the protein abundance within each organelle. So I just showed you that lysosomes are representing roughly 0.2% of the total proteome in the cell. Even still, the proteins that are present in the lysosome, as you can see in this histogram, have a very large dynamic range. So it's not that the protein abundance of each protein in a distinct organelle is similar. They actually can vary quite a large amount across the proteome. So the lysosome is an extreme example. If you look at sort of these molecular machines like the proteasome or the ribosome, you see very tight correlation of protein subunits there. So these are representative of essentially molecular complexes, whereas some of these others just have many different diverse proteins that are present in many different amounts. So another important thing that we like to try to answer in these kinds of experiments is what the fractional occupancy of any individual modification is. And so you have the potential of a 10-fold increase in occupancy of any particular modification being related to, for example, going from 0.1% to 1%, 1% to 10%, or 10% to 100%. So these are really measuring very different sort of activities within the cell if you're trying to address that level of dynamic range. And so one way that you can address this is to use essentially targeted proteomics to quantify exactly what the fraction of individual modifications are. And I'm not going to I don't have time to go through in detail, but there's ways to organize a workflow where you're using something called either parallel reaction monitoring mass spectrometry or also aqua-based proteomics. And you can measure, so for example, in this case, we're measuring in individual ubiquitination stoichiometry and abundance for a ubiquitin ligase that's modifying this protein called VDAC. And so you can use these kinds of systems to measure site specificity. So you can, in this case, measure the lysine residues that are modified, the dependence on regulatory factors upstream of those modifications, and then also convert the abundance of the modifications to the fractional occupancy by also simultaneously measuring the total abundance of the substrate, in this case, this VDAC protein. So in the last few slides, I just want to go through what I sort of see as the current challenges in the field. And so the first challenge is to understand what drives complex formation, stoichiometry, and turnover. So this has a lot to do with what Manu was talking about. How do you synthesize multi-protein complex components and have them assemble in the proper way? And so you can have this be regulated in many different ways. One way is the abundance can be regulated by the interaction partners. And so you'd sort of like to understand various aspects of synthesis rates, how different subunits come together, and what controls their turnover. So the next sort of future area that people are really sort of going towards now is how do we go beyond just a straight protein abundance to understand the status of individual assemblies. And one technique that's being used a lot now is called thermal proximity coaggregation, also calls, has other acronyms as well. But you use temperature sensitivities of protein solubility as a measure of whether or not proteins are happily existing in multi-protein complexes. And by doing this on large scale in quantitative methodologies, you can understand complexes as they change during the cell cycle, or you can look in different signaling scenarios, and also you can use small molecules to perturb these kinds of things. So these thermal proximity methodologies are now really emerging as important tools to look at this kind of question. Another question, which Anne-Claude also mentioned, is modification states. And so you can imagine you have multiple copies of one protein that is in different modification states, and these can often be combinatorial. And so there's the potential that different protein-protein interactions depend on modification states, as you see here, and that upon stimulus you can dynamically rearrange these. And so here we're going from a situation where you have different forms to essentially all the molecules in the cell having the same form. And so in this scenario, we would like to know what the overall stoichiometry and modifications are, what the fraction of each pool of the different proteoforms are, and how the modification state affects the interaction partners in complex stoichiometry, and ultimately how perturbations affect the balance. So one way we are starting to use other kinds of proteomic approaches to look at these questions is the idea of top-down proteomics, where you're not creating tryptic peptides which dissociate the individual modifications from one another. Instead, you're looking at larger fragments with the top-down methodology, where in this context, you're looking at combinatorial modifications on the same protein molecule under different conditions. So top-down proteomics is becoming a useful approach, although it does have certain limitations, such as the amount of protein you need to do the experiments. And then finally, understanding quantitative changes in cellular organization. So now if you combine organelle separation approaches with quantitative proteomics, you can get a view of how molecules are changing their localization in the cell from a quantitative sense. And so this is an example, again, from the Borner lab, where they're looking at EGFR-dependent changes in the composition of different organelles. And you can sort of get a feel for how many molecules are actually going from the plasma membrane to the endosome and ultimately to the lysosome, or undergoing different kinds of location changes in response to this, this growth factor signaling pathway. This is also possible to look at with APEX or BioID type approaches, as Anne-Claude talked about. And so I'll stop there and let Ivan talk.
Thank you, Wade. Good. So I'm gonna follow up on the protein degradation topic. It might sound as a very, very destructive topic, but in reality, it's essential for life. And you have heard the beautiful presentation from Manu, Claude, and Wade showing how the proteome is remodeled, being built, having different functions, communicate, and how we can monitor it. But if the final step, degradation, does not really operate, there is a complete clog up of the system and the cause of the disease development, aging, and many other processes. Therefore, degradation is as essential as, for example, building or functions of proteins. And I'll just focus today on the two main degradation routes to which proteins need to go to be remodeled and degraded. First of all is the mega Dalton machine proteasome, which takes the normal proteins, polypeptides, everything that's unfolded, and gets through the proteasome. And it is estimated that 80% of all the proteome goes and gets degraded via proteasome. Yet another route is the lysosome, which is an organelle rich of different proteolytic enzymes where the transmembrane receptors, protein aggregates, or external proteins via these different routes can be delivered and eventually succumb to degradation. So let me just focus now on a proteasome just for the few moments and introduce the main players and then ask the questions what are the currently ongoing. So one of the first discovered a signal for proteolytical degradation is a small molecule ubiquitin, a pioneering work from Aaron Ciechanover and Abraham Hershko. But that protein is then handled by the set of the enzyme E1, E2, and E3 such that they can recognize a substrate protein and then build up the either monoubiquitin or chains of ubiquitins on the substrate protein. And then this ubiquitin tag is the signal that is recognized by the cleavage machine, the proteasome. And here's the middle part of the proteasome, which is the catalytic site in the middle of the barrel. And then you have these two regulatory parts, which are both from both sides, so it can be capping from one and the second side. And here is where the recognition with ubiquitin and the substrate protein is mediated. And you can see this beautiful work, which is summary of many labs, including Goldberg's lab, Dan Findley's lab, Keiji Tanaka's lab, and in particular this beautiful cryo-EM imaging from Wolfgang Baumeister's lab. But you can see that today on the proteasome, we can recognize the different conformations and all the atomic details of the entire regulatory part and entire cleavage part. And then you can see how they are changing from the ground inactive state, commitment state, substrate engaging state, and degradation. And this knowledge is truly, in the last few years, a total revolution in full understanding how ubiquitin communicates with proteasome and how proteasome can degrade a variety of different proteins. So here are a few interesting facts which I believe will attract some of the attention of the audience and go for the question. We talk a lot about numbers and we heard about from Wade quite a lot. It is estimated that around 1% of the total proteomes are proteasomes. And this accounts approximately 1 million proteasome units per HeLa cell. And then you can really monitor them by this beautiful method of the cryo-EM tomography. You can see where they are localized in a cell, on the nuclear envelope, organelle, cytosol, and so on. And not only that, but you can also look how they are, in which conformation they are, whether they are double capped, whether they are in the active or inactive conformation with a different percentage at a different stage of the cell. So the knowledge that we have today is truly much deeper than previous, just drawing the model of proteasome. And then there is a very ongoing and very active research on what are the optimal ubiquitin degradation signals. Is that only K48, as originally worked by late Cécile Picard, showing that the 4 tetraubiquitin on K48 is predominant signal. Today, we know by work from Aaron Ciechanover and many others that monoubiquitin can serve as a efficient signal. Different type of ubiquitin chains, and particularly, recently the work of Mihai Rape, the branch K48 and K11 chain seems to be the most competent or capable of degrading in a fast manner, for example, proteins from cell cycle or aggregated proteins that cause neurodegenerative disease. Then one of the actually a nice number play. There is a web page called Cell Biology by the Numbers. You can actually calculate the speed of the ribosome polymerization of proteins and proteasome degradation of proteins. And you're going to come out to very similar numbers, such that there is always a balancing. And when you calculate, you can see that per day, the entire proteome of the cell can be degraded by the proteasome, and this requires 24 hours. And very similarly, the numbers come out for the ribosome synthesis of the full proteome back. And we talked quite a lot about technologies, and in the technologies as well important in monitoring ubiquitination, particularly by utilizing the mass spec approaches we heard from Wade. Then also protein degradation. There are many different methods, GPS, we can look for ubiquitin sensors, we can utilize specific antibodies which recognize K48, K11 branch chains, and so on. There is also a different type of ubiquitin probes where you can capture all of these acts. And this is quite an active part of the research where we are trying to delineate what are the optimal substrate which can actually be degraded in seconds from those which can be ubiquitinated but degrade very slowly. And along the technology side, I really would like to emphasize again the cryo-EM tomography, particular work of Baumeister, which tells us where they are localized, what are their conformation, what is their activity level. So as everything in cell, it's also localized. So one can also look to the neurons and look where the proteasome in the somi, or whether they are in axons or dendrites. And you can see that both ribosomes and the proteasomes are found in a very small dendritic spines, and locally they perform full function of translation and degradation in very coordinated manner. This is like a small microcosmos here of the quality control which need to be preserved such that the synapse can work. And then again, you can also look what happens when the neuron is diseased. For example, with these poly-GA aggregates in C9orf mutation which causes amyotrophic lateral sclerosis, you can see that these aggregates can go in a direct contact with the proteasome. They may recruit them and may inactivate them such to block the effect. And the consequence of that is then that these aggregates cannot be removed by the proteasome. So just one last topic on a proteasome research. It's already known that there are few drugs in the clinic which block the catalytic site of the proteasome. But very exciting new part of the research, which was originally pioneered by Craig Cruz and Ray Deshay, is called PROTAC. PROTAC is actually a small engineering molecule which can bring together a specific substrate of our interest that we want to degrade with a ligase which will add ubiquitin chains, and that will be the signal to be degraded in a proteasome. And by utilizing the small chemical synthesis or medicinal chemistry, one can develop today a variety of PROTACs which can have a wanted effect. We can degrade specific oncogenes of our interest via the proteasome degradation. And that's something, again, which opens quite a lot of new areas in the research in a recent time. So as I was alluding to you, the protein aggregates cannot be dealt with the proteasome. They are dealt by lysosome. A lysosome is an organelle, as shown here, which can be fed by phagocytosis, by autophagy, or by endocytosis. And today I'm going to focus mostly on protein aggregates and autophagic pathway, how they come to the lysosome. This comes out from many different studies which have shown that there is a very active crosstalk and interplay between the proteasome pathways that are linked to autophagy. And when you block proteasome by mutations or by the drugs or by inhibitors, the consequence is a variety of those for the cell, but in most of the cases, you have always activation of autophagy as a response to the deficient proteasome function. So it's a backup for anything that cannot be dealt by proteasome. And autophagy is a cellular phenomena that cell can create these double-layered membranes which can surround the protein aggregate and deliver them to the lysosome or to vacuole in yeast cells, as it was a pioneering work of Yoshinori Osumi's lab. And if you look a little bit to autophagy, it is a phenomenal pathway by which you build up these very aggressive membranes which have an ability to eat. They can eat organelles, but they also can eat protein aggregates, degrade them in the lysosome. And for that, The autophagy is truly one of the major quality control pathways, and it's estimated that approximately 15% of the proteome is at the steady state eaten by autophagy instead of proteasome. So we know a lot about autophagy. Genetic screening yeast have shown that around 35 core genes, but you can see that there are many different pathways, from the kinases, PI3 kinase, remodulation of membranes. But what is very critical, that there is a conjugation of these ATG-like molecules to the membranes, and this makes them aggressive. And once they have these protein modifiers, these modifiers can actually engage and recognize protein aggregate to eat it. And therefore, we can understand that there is a very basal upon starvation, and there is a selective aspect of the entire autophagy pathway. And the selectivity is understood today much better, and this is one of the very active part of the research in many labs across the world, in which we understand now that different cargoes might be recognized by specific selective receptors, which will eventually bring these double membrane autophagosomes around the cargo, let it be protein aggregate, mitochondria, or ER. So the concept that Alexandra and Andreas and myself, we put forward is that actually selectivity is defined by this subset of different receptors. And very early work from Terry Johansson, Masato's lab, have actually shown that the protein aggregates can be recognized by the P62, an adaptive receptor protein, as well as MBR1, and that these will actually bring them to the autophagosome for the full degradation. And you can immediately notice that many of these receptors also contain the ubiquitin binding surfaces, such that ubiquitin is very often a signal on a cargo protein aggregate to be degraded. So let me just stop now by just leaving you with a few ongoing challenges in the field. First of all, we'll understand yet very little about the kinetics of degradation of individual proteins, why some proteins are second-based and other ones are hours or days. Then there is a lot of coordination of proteasome and autophagy systems, which we are still not understanding in full capacity. That's one of the very, very active areas. And the issues of local degradation and competition between different substrates for proteasome versus autophagosome also raises quite an important questions. And most of this, all this biochemical knowledge that we come together by studying proteins, has a clear clinical relevance of both systems, or all the system from building proteins to degrading them, because they're relevant for the development of human disease, but they're also relevant because the new therapies are utilizing those systems to fix the damage and to give the healthy life for our cells. Thank you very much for the attention. So maybe I would also now stop by saying that I would like to thank Manu, Wade, and Claude for really great presentations. Now we are open for a discussion panel, and maybe Cora from the eLife can actually put forward any questions that came.
Yes, hello, thank you, Ivan. So we've got a couple of questions here. First one from Joanna Zawadzka-Pankau. What method would you recommend to detect a conformation change in the intrinsically distorted proteins? So I'm not quite sure who that's addressed to, but I presume one of the proteomics guys would have insight into that.
Sorry. Yeah, so this is actually hard, right? Detecting conformation change in intrinsically disordered protein is not an easy task. I mean, we have been dealing with such question in the past, and I have to say we tend to work with NMR people to do this kind of work, but also in terms of proteomics, I mean, if you get a decent, I mean, if it's completely disordered and you're alone in solution, a change will not be picked up by any of the approach that we're using. If, however, you're looking at a change upon binding to something, then you can do something like either HD exchange or any of the approaches that will be exploiting, for example, differential susceptibility to cleavage by a proteolytic enzyme. A little bit of the work that Paola Picotti, for example, has been doing, and maybe Wade has other idea.
I mean, there are also the chemical probes now which are used, or different small chemical compounds which are actually recognizing conformational changes of the active complex. And this, even in vivo, at the steady state endogenous complexes, can be very, very meaningful and capture the stuff. In a crystallography aspect, obviously, nanobodies, different type of antibodies are used to stabilize the conformation and then solve it. And obviously, cryo-EM there is having a great impact.
I don't have anything to add.
So maybe for you, Wei, there was one question here on quantitative numbers. It says here, given that 50% of protein represents only 2% of proteome of the cell, is it likely that most of those that are involved in signaling, and therefore their presence or absence in the proteasome, is temporal?
It's temporal. In other words, it's time dependent. Yeah, I mean, so one of the limitations about the numbers that we have currently, first off, it's usually on a very small subset of all the different cell types. So it would be in a commonly used tissue culture cell or something like that. And generally, they're not really looked at this level so far in any kind of dynamic situation. There are a small number of studies where dynamics is looked at, and so there's not generally very much information about the temporal abundance and how that might change. Also, where a protein happens to be in the cell might matter more than its abundance, or its modification status might matter more than its abundance. And then finally, on the sort of edge of the spectrum of very low abundance proteins, these are sometimes hard to measure doing global proteomics. And so experiments that try to get at those low abundance proteins by using enrichment strategies, which is, you know, in reality what most people do when they do an IP, they're enriching for a protein that they want to look at that's maybe of low abundance. And so by incorporating enrichment strategies, you can gain access to those much lower abundance proteins. I don't know if Anne-Claude has more to add.
No, actually my sound cut and I missed most of your answers. I'm not going to repeat, but the end of it made sense. Yeah.
Yep. I hope the audience have heard all of your answer, Wade. That was important. So because we are a little bit short of time, I think we are getting a sign that we should maybe invite everyone else to ask and contact us directly if there are any questions from what we presented. I think it's an open forum for sharing the information, particularly the new technologies and the use for different biological questions. I would like to thank once again the panelists, Wade Harper, Manu Hegde, and Anne-Claude Gingras for being with us today, for sharing their knowledge and sharing the enthusiasm why the protein biochemistry is still very hot and very important in life sciences. Thank you all for participating, particularly to people who have joined us and who have sent the questions.
All right, my pleasure.
Thank you.
Thank you, and thanks also to Cora for coordinating.